pleasure to welcome Pietro Spanu to give his inaugural lecture. Pietro did his PhD and a short postdoc at the Friedrich Miescher Institute in Basel. And then in 1992, he was awarded a Royal Society University Research Fellowship, which he took up at the Department of Plant Sciences in Oxford, and he was there for 7 years. And then in 1999, he was appointed to a lectureship in the then Department of Biological Sciences, which subsequently morphed into the Department of Life Sciences, and was subsequently promoted through senior lecturer and reader, and now to professor. So this is his inaugural lecture, which is entitled Hackers at the Dinner Table. Pietro.
So let me get this working, if I can. Okay. Well, first of all, welcome everybody, and thank you very much for coming. I am delighted to see so many of you from quite far as well. As everybody from just across the road. So these, for those of you who don't know, these inaugural lectures, at least as far as I understand, are a way of celebrating this sort of event, accession to professorship, with a talk which is supposed to be aimed at a general public as well as perhaps more specialized colleagues. This is a huge challenge, and I'm very jittery because it's a very unusual sort of lecture. So let's hope that it works okay. Hackers at the dinner table. Well, what is this about? Well, this is about what has been the path of my research in the last few years. And I want to tell you something of an idea that has sort of dawned on me in the last few months that I think is quite important and significant and is exciting me. And I will try to explain what I mean by this in the first half of the talk, and then the second half I will go into the specifics of my own research. Okay, so hackers at the dinner table. What do I mean by the dinner table? This is our standard evening meal, okay, or the table prepared for a standard evening meal. When we come to our food, we often forget that, in fact, we share our food with others, okay? And these others are not just the guests or members of the family, but there are other organisms that make bids for our food and our drink. Okay, we conveniently forget this because we live in affluent, secure, reasonably healthy societies. But behind the scenes, okay, there's this. Most of our food actually comes from plants, and even when we eat animals, for those of us who are not vegetarians, the animal food comes from plants. And plants, you can see these in an idyllic setting, are always prone to being attacked by all sorts of microorganisms. Some of these microorganisms are actually beneficial. Most of the ones that I've worked with, the beneficial ones are present in the soil, and they help plants to grow, but I won't be talking about these. I'll be talking about the ones that we actually compete with, or let's say they compete with us. These microorganisms cause plant diseases. And I'll spend a few slides here to remind those of you who know all about plant diseases some important points, and perhaps introduce some of these to those of you who aren't so aware of them. And the first one I want to talk about is a microorganism that pretends to be a fungus. Okay, it's important. It's not actually a fungus. We call it that, Phytophthora infestans. It grows like a fungus on a petri dish. It makes spores that can disseminate and can infect plants. And let's see if this works. This is a little video that a colleague in Norwich produced. There's a bit of music going on behind that.
Here's a scary parasite with flagellated spores and hyphal threads.
Okay, and that's all you're going to hear. Okay. I do actually recommend that I've sent it around colleagues here in the department. I know that they've been spammed already, but do have a look. It's a very short video, but it's very instructive. Okay, it contains some very important points about this plant disease that I will talk about here. I actually worked with this at the start of my PhD, but I'm not going to talk about those results here. So, Phytophthora infestans causes something called late potato blight. Okay, and if you inoculate potatoes by spraying those spores on the leaves, after a few days you start seeing shoot dying back, and fairly rapidly, so within about a week, the whole of the plant dies. Now, it's quite dramatic. And you can imagine, if that happens, well, what's going to happen to the potatoes that we are really interested in? We don't eat, obviously, the green bits. Well, what happens to the potatoes is that the spores from these parts here land on the soil, and they attack the tubers as well. Okay. Now, this is what happens to a potato field inoculated with Phytophthora. That looks reasonably healthy. After a week, that's what happens. And this is a very real pathogen with serious consequences. Again, we tend to forget about this, but that's what happens to the potato. And there are all sorts of other microorganisms that cause a secondary attack and essentially degrade your potatoes, and you've got nothing left. Again, we tend to forget about this. But if you are totally dependent on potatoes, like a very large portion of the Irish population were in the middle of the last century, you have nothing left to eat, your poverty is exacerbated, and you get evicted from your home. And as probably most of you know, this was the result of this coincidence of social and economic situation and the devastation caused by the blight really resulted in many millions of Irish starving, dying. The lucky ones, the ones who had a little bit of money left, managed to emigrate to the United States. The unlucky ones emigrated to England or Scotland. Okay, and this was terrible. I mean, this was terrible. Now, again, this is another century, different population. We forget about this thing, but I don't know if any of you recognize this photo. This is only about 1 kilometer from here. Okay, and again, we sort of forget that this was happening very recently here. We've moved on. Of course, all of this wasn't caused directly by Phytophthora infestans, but plant diseases can have impacts like that. Now, Phytophthora is a disease which causes complete destruction of the crop, but fungi and fungal-like organisms can do other interesting things to crops. For example, this one here, Claviceps purpurea, causes ergot. Again, it's a disease that we tend to forget. It's a disease of the past. The fungus produced these weird fruiting bodies on the heads of cereals. And the problem there is that these fruiting bodies, or this sclerotia, look very much like cereal kernels. This is barley, which is the plant I work with. And you can see these brown, dark brown sclerotia mixed up in the barley. And they contain some very powerful toxins. These toxins cause gangrene and convulsions, and that is what we called ergotism. Okay, and this was very common in Europe throughout the Middle Ages and after that. There are lots of depictions of people affected by gangrenes and convulsions. There are some extremely interesting connections between ergotism and various social events. There are some theories that connect ergotism with witches' trials, for example, in Salem, and so on. There's some debate about that. Again, we tend to think of these diseases as something of the past, something we are not affected by. But that is not such a long time ago. This is an example of gangrene caused by ergotism in Ethiopia, only 15 years ago. This is a well-described outbreak caused by an outbreak of Claviceps purpurea in Ethiopia. Now, other fungi, like the ones that I'm working with, don't produce toxins. They don't destroy the plant. They just take up nutrients. So they really share the table with us. But they compete for nutrients, and if these fungi infect plants, the yields are reduced to make the crops not very valuable. Now, what do plants do in response to this? They don't, well, yes, they do just sit there. Plants, as you know, most plants don't move, but they sit there and they try to defend themselves. And throughout evolution, plants have acquired the ability to produce a vast array of chemicals. Essentially, these are their own antibiotics. And those of you who are interested in chemistry can look up all the diverse molecules. Here is elemental sulfur, for example. And these are quite effective against attacking microbes. We call these phytolexins. But plants, yes, they sit there, and you will see in this little movie, these are the cell walls. Don't know if you can see them here in red. These are plant cell walls that constrain the plants. That's one of the reasons why plants don't move and plant cells don't move around. But, whoops, that wasn't supposed to happen. That also wasn't supposed to happen. Let's see if this is any better. No. Okay. So here you have a plant that's infected with Phytophthora. The Phytophthora are these red circles here. And if you just focus on this part here, or on this part here, you'll see that there are things moving towards the Phytophthora. And then after about 40 minutes, there's a big vesicle that explodes and results in eventually the disappearance of the structure. Okay? And this plant vesicle that was there up to about 40 minutes contains an array of enzymes which essentially digests the cell wall and the Phytophthora. So these are another set of defense mechanisms that plants can deploy. And we know about this because breeders have, throughout the last century or so, managed to select plants that resist attack of certain strains of Phytophthora. Okay? Takes a little while to develop these strains, but these strains are resistant at least for a while, against diseases. And they are one of the pillars that supports our, let's say, safe, secure food supply. Okay. There are some problems with this. Phytophthora is very good at evolving, and so if you have whole crops made up of a disease-resistant plant, the Phytophthora will eventually evolve and, let's say, learn, in inverted commas, to infect, and it will break down the resistance. And we see this over and over again. So agronomists, breeders are in a never-ending race to try to keep up with the evolution of the pathogens. Okay. Now, we have found out that plants are very good at perceiving microbes when they attack them. They have areoles that look a little bit like these. Okay, they're very sensitive. Oops, sorry, wrong one. And these areoles are capable of perceiving signals that come from the pathogens. And they then convert these signals in messages that will then go to the control room of the cell. Now, this is obviously a radar. It is an analogy. But this is what the plant receptors really look like. This is a cartoon, a simplified cartoon. And I've taken this cartoon, which was published in a paper by Thomas Buller and Jörg Felix, because Thomas Buller and Jörg Felix were my PhD supervisor and senior postdoc, to whom I owe a great deal. And in fact, when I started my PhD in Switzerland with them, they had just started working on this project. And it was all very exciting, except that I was there about 7 years too soon. The discoveries, these very exciting discoveries, were, let's say, made after I'd left. But in essence, most plants have these molecules that really look like this, that are like areoles ready to perceive signals made by microbes here. And as a result of this perception, they then initiate what we call the defense program. Okay? That is a molecular structure of this receptor called FLS2, at least part of it, the extracellular portion. This is a co-receptor, and here in pink you have the image of the piece of the bacterial molecule that is perceived. Intriguingly, animals have got very similar receptors. For those of you who are interested, the toll-like receptor 5 is an analogous structure to this, and it also perceives bacterial flagellin, but a different part of bacterial flagellin. We think that animals and plants have converged into evolving this particular reception mechanism. And what happens when the plant perceives these signals is shown here on the right. A whole set of what we call signal transduction mechanisms inside the cell are triggered. Again, it's very complicated. This is a gross simplification of what is really going on. But those of you who are going to have who have good memory should note the centrality of this MAP kinase unit, which then triggers gene expression and expression of the defense program. Okay, so this is now a cartoon of what we really think is going on in plants. So, importantly, plants have got essentially a burglar alarm, and inside the cells they've got a modular burglar alarm. And if you are a microbe, a pathogen, who's interested in entering the plant, you need to have tools. You need to have tools to break in. and you need to have tools to interfere with the burglar alarm. Okay, and this is indeed what we've discovered. Microbes do have these tools. Okay, and most of these tools are proteins that we know of, are proteins. And proteins, for those of you who are non-biologists, and please bear with me, are made more or less like this. Okay. Some of the proteins that the plant makes will then activate defense here. Now, I don't know if you can see this very well from that. Let's see if this writes a bit better. So the program of the cell is encoded by DNA. I think most of you probably know this. The information in the DNA is converted into RNA, and then RNA is translated into proteins. Now, this has been known for, 70-odd years now, 60-odd years. Perhaps more recently, we've found out that RNA does lots of more interesting things, including regulating its own existence and synthesis. Okay? And this is the real exciting thing that's come up in the last few years. Some pathogens, okay, some microbial pathogens, some of these fungi and fungal-like organisms have learned how to interfere with these processes here. So let's say, this doesn't write. Okay, this is a wall. Here we have the pathogen. And here we have the plant. And in this paper, published a couple of years ago, this is not my work, the authors have found that the oomycetes, this is Phytophthora, the potato blight, can interfere with a process called RNA silencing. Okay? Now, the experiment here is a little bit complicated for those of you who don't do these things every day, but the results are shown here. In the absence of the oomycete molecule, you have a silent leaf that is not green, it's red, shown here. If you then introduce these proteins called effectors, PSR1 and PSR2, into the leaf, you can see there's a green patch appearing. Okay? And that is indicative of the fact that the pathogen has silenced the silencing. Okay, this is a bit complicated. Two negatives, that gives you a positive signal. Okay, so these are PSR. And, well, that's interesting, but what has this got to do with disease? Well, what has this got to do with disease is shown below. In these plants essentially become more susceptible to infection. So as a result of the silencing, you get a more susceptible plant. Okay. In this other paper, something even more interesting has been discovered. This other pathogen, this is botrytis. This is a true fungus in this case. It's a pathogen which attacks lots of things, including grapevine. So it affects our drink, not necessarily our food, although it does that as well. What botrytis does, it actually sends in some RNA, okay, which itself is doing the silencing, okay? And this is new. This is completely new. An event where a eukaryotic microbe is targeting some ribonuclease, ribonucleic acid, okay, to affect essentially the function of the host cell. Okay. Interestingly, this process is dependent on the plant's own silencing mechanism. Okay. And this is what I call hacking, because what we have here is essentially a pathogen that is getting into the computer, okay, and affecting the software, the mechanism of transmission of information. Okay. And so this is what we're seeing, okay? And it's quite new. Okay, now, step back, and where do I come in? Well, this is where I come in. This is where I start. A long time ago, when I was young, etc., I grew up on a diet of these books, My Family and Other Animals, and Conrad Lawrence, and related texts. In fact, I was a committed zoologist. Okay, I really wanted to study animal behavior, like all the other students who come here and study biology. Except that when I got to university, there were two problems. A, I wasn't very good at zoology, so I got low marks. And B, I really didn't get on with the zoologists. Okay, and that's an understatement. Fortunately for me, I was really fascinated by the plant biology faculty at my undergraduate university. And in my second year, this was in 1983, somebody who was a researcher there organized a symposium and asked the students to do some reading and present something about the serial endosymbiontic theory for the evolution of organelles. So I bought this book and I read through it and I was just hooked. It was fantastic. I don't know how many of you have read this, but most of the biologists here probably know about the theory. And I became fascinated about this idea that microorganisms could get inside other cells and essentially evolve into very interesting things. This was in 1983, second year biology. By the time I had a short interruption of studies as a CEO. I didn't want to do military service in Italy. I came back, and the faculty had organized a conference where they'd invited the author to speak, Lynn Margulis. She's very charismatic. I don't know if any of you had ever heard her speak. She was amazing. And so I was hooked, and I wanted to work on symbioses. And so I started my undergraduate research project there. I didn't realize at the time, but it was very fruitful. We got 4 papers published out of my undergraduate research project. It's never happened again. I've never been so productive. And also, I felt completely depressed. I thought it was awful. I thought it was a waste of time. So how wrong can you be? Interestingly, one of those papers, I just highlight here in this journal, I thought it was an obscure journal. I mean, look at the font. It looks really antiquated. Now, New Phytologist is one of the most difficult plant journals to get in, and I'm on the editorial board now as well. But here, in this particular work, I found that in mycorrhiza, okay, these are beneficial symbioses, we could observe a reaction on the part of the plant. So the plant was responding as if it were a pathogen, in fact, and that was all very unusual. So that's why we managed to publish it. But let's move on 20 or so years. Just before I came to Imperial, in fact, I started working with cereal powdery mildews. And the reason why I worked with cereal powdery mildews is because I was very interested in symbioses, and cereal powdery mildews cause this sort of disease. And it's really because of this, that this photograph, this image that's imprinted in my imagination, that I came to this. Here you have an infected leaf that, apart from the fact that it's showing all this powder, which is the spores of the fungi, the leaf is green, okay? And this is actually a puzzle. How can you get a pathogen that's infecting a plant and the plant is alive, looks happy? It isn't really. All the food is going to the fungus here. So this plant is not going to produce much in the way of wheat or barley. Now, I work on the barley powdery mildew, but there's a close relative, the wheat powdery mildew, that does more or less the same thing. You know that wheat is important. Everybody knows that wheat is important. I think it was first published in this paper here. Don't know what the impact factor is, but you know that it's not ref returnable, right? Okay? Not ref returnable. It's a review. It's not a primary publication, so not really important. But what you might not know is that barley is very important as well. And seriously, the barley acreage in the UK is only about half of, it's as much as, sorry, as half of the acreage of the wheat in the UK. And this is increasing, in fact, in other countries as well. Many of you may know that barley is used to make beer, an essential component of our diet. It's also used to make the most precious, valuable export from the UK, agricultural export. Okay, well, as long as the Scots remain in the UK, I think that is. But perhaps more importantly for food and for the theme of the talk, barley is used as feed for animals, which we then go on to eat, or in more recent years has been found to be very good for sort of as a sort of supplement to foods itself. Okay. Now the powdery mildew, how does the powdery mildew infect? Well, it produces spores that look like this, and as soon as the spores land on a surface, they produce a tiny hypha that looks like a tongue. It just looks like a tongue, but it's actually working like a tongue. It tastes the surface, and if the surface is the right one, it then produces a second germ tube, which is very different from the first one, and this germ tube then enables the fungus to penetrate that surface. And if that surface is a barley leaf or a wheat leaf, depending on which variety of the fungus we're talking about, inside the cell, they'll produce this thing here. This thing is, we call it a haustorium. Essentially, it's like a pair of hands with fingers, and these grab nutrients. They literally grab up the nutrients, and they transfer then the nutrients from inside the plant cell to outside where the fungus is growing and making all those spores that you've seen. And this picture, which Micah knows very well, has been iconic for me, again illustrates why this is so fascinating. The plant cells are completely filled with these structures. Now, these plant cells, amazingly, are alive. They are completely alive. And these are plant cells that have got all these perception mechanisms. So how can they have been fooled to ignore the fact that they are full of pathogens? And so this is where we started from, and this is where I've been working on. Okay. If the fungus is successful at infecting the leaf, it will then grow on the surface and produce all of these spores, which then within 5 days go on and produce more infection. So the pathogen itself is extremely active as a disease-causing organism. Now, there's a slight problem with these fungi. We can't actually grow them on a petri dish, and this causes all sorts of difficulties. However, in the last decade, as a lot of you know, the ability to sequence genomes sort of burst in remarkable and perhaps in a way which we didn't predict. And we were able, finally, thank you, Nick, to get some funds to sequence the genome of the powdery mildews. And we had hoped that this would give us essentially an instruction manual for the fungus itself. Well, this sort of worked out. The actual cover of the manual, when we finally got to publication, looked more pretty. Okay? And although this is not powdery mildew, this is downy mildew, we went in with a raft of other publications in that number. And of course, at that stage, we were all very pleased. But now I'm going to tell you what we found and why it's important. Well, we found lots of surprises. Okay, that's a positive way of spinning the fact that we found what we didn't expect. First of all, we set out quite sure to find a small genome. This is because the biologists know that in parasites, especially obligate parasites, tend to have reduced structures, smaller genomes. Okay, and we were wrong, very wrong. Okay, It caused a lot of sweat, especially because we'd used up most of the budget to sequence something that we thought was very small, in fact was huge. How huge? Well, about 4 times the average fungal genome, and a lot more than we expected. And all of the powdery mildews seem to be like that. The second surprise was that when we actually started decoding, reading that instruction booklet, we found that most of the primary metabolic pathways, and practically all of them, are represented. We were expecting that all of these metabolic pathways were, at least most of them, would be somehow disappeared because the fungus, we can't grow the fungus on a Petri dish. But in fact, most of them are there. Okay. The third surprise is, well, it's a surprise because after we found such a big genome, we thought, well, there's going to be lots and lots of genes. Well, no, there's only about half of the genes that you find in other related fungi. So that and the genome size means that the gene density of the powdery mildews is very low. Okay. Now, why are there very few genes, or what are the genes that are missing? Well, interestingly, what happens is that the gene families tend to be very reduced. There are very few paralogs. Fungi, in general, have some very large gene families of certain sets of genes, for example, genes encoding enzymes of secondary metabolism. These are related fungi, and you can see there's lots of different types. Blumeria, which is the powdery mildew fungus, only has 2 of these. Okay. The same applies to enzymes, genes encoding enzymes for breaking down the cell walls. Related fungi have got lots of different versions of these. Blumeria, the powdery mildew fungus, has only got 1 or 2. Okay, so these tools, they have tools to get in the plant, but there's only just 1 or 2, and that was a bit surprising. But what was most surprising is that we found a huge superfamily of genes that encode things that we call effectors. And these effectors are those tools that I talked about, tools to enter and do things to the plant's immune system. And so you have to see this in the background, that most of the other gene families are restricted, so there's hardly anything there, whereas the effectors seem to have sort of multiplied ad infinitum. Well, not ad infinitum, but there's 530 of them, which makes about 7% of the total gene coding capacity of the fungus. And these genes are not just doing nothing, they're expressed, and interestingly, they are expressed at high levels in these haustoria. Okay, this is the blue histograms here, and higher levels than outside the plant. And they are not just producing RNA, they're producing proteins. Thank you, Laurence. You found these. Okay, wherever there's a dot, those are proteins that we've found. So they're really real genes, and they're making real proteins. And they're upregulated in the haustoria. And not only are they upregulated in the haustoria, they've got something else very interesting about them. If you analyze carefully the sequences and you compare the families within the candidate effector superfamily, you can see that some of the families, gene families, have got a very high ratio of the non-synonymous to synonymous mutations. Now, for the non-initiated, this means that these genes here have been subjected to very strong diversifying selection. So in evolution, there's been a pressure to diversify these genes. Okay? So these genes multiply, there's lots of them, and then they start becoming very different. And we think this is driven by the plant. So, summarizing, when you go into a fungal genome, you see a flat or a room that looks like this with all sorts of knickknacks and things that are nice. But when we went into the powdery mildew genome, we had a minimalist genome. Everything necessary is there, but not very much of it. And in the cellar of that minimalist genome, we found a toolbox with lots of tools. And this is extremely exciting, and that's why I've been working on effectors, on these effector genes ever since. Really, the question is, what are these tools doing? Okay. And to do this, we took a sample of those tools. We took 50 effectors that are very highly expressed as proteins, and we silenced them to see whether they were essential for pathogenicity using something called Higgs, host-induced gene silencing. We measured the rate of formation of Haustoria in samples that we had silenced compared to those that we hadn't. Essentially do this long boring screen by Clara. Some of you might remember Clara here in the department. She found that, okay, you get a range of responses, but some of the genes here seem to be very important. When you turn them down, you get less pathogenic development. And arbitrarily, we just decided to focus on the first 8. We call these BECs. Okay, Celia knows these very well. BEC 19, Celia is the protein that Mum and Dad always argue about. Okay, so I'm not going to talk about that today. I'm going to talk about these two here, that are the ones that are closer to my heart, BEC 11 and BEC 54. Why am I looking at these? Because apart from the fact that they give the strongest response, they're related to each other by sequence. However, their Ks ratio is extremely high. If you have a ratio of around 1, you think there's neutral selection. Anything bigger than 1 is diversifying selection. Normally, something around 1.2, 1.3, 1.5 is indicative of strong selection. This is nearly 4. I mean, it's massive. And if you actually compare the sequence of the primary sequence of the two genes, I haven't got time to go into details here, you can see that there's great diversification in the exons, which really confirms this structure. Another thing that we have observed is that these effector proteins and effector genes are turned up very strongly immediately after germination when the fungus is growing on the plant. You can see here the RNA profiles, and this is essentially fold induction relative to conidium, so it's in the thousands. This is very remarkable. Thank you, Lee and Helen, for doing these experiments. And now the really interesting bit. If we put these genes into plants, okay, completely different plants, the plants become more susceptible to disease. And we've done this with wheat, okay, the transgenic wheat, and you need to compare these bars here, this one to this one to this, and this, etc. Okay, the transgenic plants are more susceptible to to a pathogen that is adapted to grow on wheat, but it's more susceptible. And the same applies to Nicotiana, which is a tobacco relative. Okay, and again, we can induce susceptibility. So we have introduced the tool for pathogenicity, the effector, in the plant, and the plant becomes more susceptible to disease. And this is extremely important for us. And another thing really makes me interested in these genes is that they look like ribonucleases. We call them RALF genes. RALF is a backronym. Okay, RALF is actually my colleague in Germany who first discovered this. Okay, and so we invented an acronym that goes to his name. It's RNAs-like proteins expressed in Haustoria. It fits. Look it up on Wikipedia. I learned yesterday that this is called a backronym. Okay, and this is what we observed, or what Ralph observed, in fact, using fire. Thank you, Mike. Okay, if you predict the structure of these proteins, the structure, so the primary sequence doesn't look like an RNase, but the predicted structure looked a little bit, not very convincingly at the time, like ribonucleases. But when you start doing this over and over again, and you keep on seeing that the structures, predicted structures, match to each other, it becomes a little bit more convincing. Okay. Now, these RALF genes, these BEC11, BEC54 are down here, part of this little green family. But actually, there's lots of them. So there's about 120 in the big effector superfamily. That in itself is indicative that they're probably doing something really important. If you force an alignment of the proteins, and you really have to have faith, you can start seeing perhaps things aligning. It's not very convincing, okay? But what is amazing is that, in fact, there is an indication that they're all derived from the same gene, there's an intron which is nearly absolutely conserved in all of the Ralph genes in that position. Okay? And this is extremely unlikely to have arisen by chance. Okay? We call them RNase-like genes, but not RNases, because in fact the amino acid residues that are necessary for the catalytic activity of an RNase is missing. Okay? And in fact, when we make these proteins in E. coli, they do not have an RNase activity. So at this point, nobody was really convinced about the structure. So we approached Ernesto. I don't know where he is. Thank you, Ernesto. And he said, oh, well, this is really easy. These are small proteins. We can determine the structure. We can ask a brilliant graduate student of mine to do it in her spare time, okay? Which she did, okay? I know for structural biologists this is peanuts, but for me it was amazing. I mean, this is the structure of Bec54. This is now the real structure, experimentally derived structure. And I hope you can see the similarity with RNases. They're not exactly the same, but they really do look the same. Okay? So these are RNases that are not RNases anymore. What are they doing? Are they perhaps interacting with RNA? Well, yes. And we've found a whole series of, let's say, bits of evidence that each individually were sort of perhaps not so convincing until perhaps this one here, that again Rianne in Ernesto's lab has recently found. If you do an NMR analysis of Bec54, you see a nice signal. These are these spots in black. And if you then add nucleic acid to the protein, some of these spots shift. Okay? These are not vast shifts, but they're marked shift. Some of them don't. And this enables Rianne to determine that these amino acids seem to be interacting, touching the nucleic acid. And if you look at the real RNAs and the amino acids that are in contact with the substrate, with the RNA here, these are the ones that are necessary. So they're not the same, they just look in the same position, and they may be doing the same thing. So this is work in progress. Of course, the big question is what is actually sitting there, okay? And this is where we are at the moment, okay? Now, my excitement with regards to hacking is I think that we have the evidence that that big toolbox we've discovered in the powdery mildew genome is actually the toolbox of a hacker. Okay, it's got 120 genes that have got something to do with RNA. We don't know what this something to do is, if it's something to silence where these proteins are delivered to the plant to silence something important there, or whether they are carrying bits of RNA. That's also possible. And we're trying to find this out. Okay. And ideally, in the sort of distant time, somebody, not myself, I'm sure, but somebody might use this information to develop some antiviral software, essentially to prevent hackers from attacking the plants, so that we can then have this idyllic scenery. I should have the violins playing here. This is from Syngenta. You know, it always looks nice. Okay, but that's what we're aiming at. And of course, this will enable us to continue having a dinner table, which we can share with our friends, family, and perhaps a little bit less with the pathogens. So thank you very much. The most difficult part of this talk is that I don't have enough time and I will send you to sleep, but I don't have enough time to explain and thank all of those who've contributed to this work and to my work. This is, all of this has been done by, literally hundreds of people. I mean, only on the genome paper we got 63, I think, authors just on one paper. And we've worked together, and that's just for one aspect of the work. So I've just selected a few that I want to thank here. I wanted to thank my two supervisors. Paolo Bonfante is a mycorrhizologist who was so fascinating when I was an undergraduate, and we've worked together, we stayed in touch, and unfortunately can't be here. Thomas Buller, my PhD supervisor, who saved me, gave me a job, became my first paid job as a scientist, and set me on the right road. Nick here, you'll hear from Nick in a minute, he's representative, these people are just representative of others. He's representative of the colleagues here in the UK who've supported me throughout these years. We've worked together at various stages on this pathogen and on other pathogens. And a special thanks to James and Sarah, who are in the audience. These are representative of the many colleagues here at Imperial who have, made my life quite pleasant, and perhaps more importantly, have made my life very productive, mostly doing work, well, for me or with me, let's say. So thank you, James, thank you, Sarah, and thank you, everybody else. I know I'm missing out people. You know who you are, and lots of them are not here, okay, but I am grateful. Obviously, special thanks to the people who've been in my group throughout the years. The PhD students, at least some of them, those who've been more involved in the work that I've been talking about, from Hannah, Maike, who's sitting in the audience, who actually has been there as a graduate student and as a postdoc, and all the others, Tim, I think he's here, and also Helen, who's a current PhD student, and Lee, who's just joined me, and Michael, who's not joined me, but is going to join me in October, okay, but has already started working on some of these aspects. Okay, so thank you to all of you. And then, of course, the postdoctoral fellows. I'm happy to see Sabina here. And Joe, who's my current postdoc, couldn't be here tonight. I also obviously want to thank my family, who's been really supportive throughout the years, all the way back from my family and other animals. I don't have time to relate anecdotes now. We can do it later on. First of all, to my father, who actually died just a couple of weeks after I was told that I'd become a professor. My mother, who's here, she has stood by me for many years and always encouraged me to do unusual things. And these are my sons, who are at university, busy doing things like coursework and exams, and Celia, who is here listening to her dad. And of course, the most important person for me here on the family side of things is Laurence, my wife, who's also a colleague. We have collaborated through the years, and I mentioned some of the work that we've done together, and hopefully we might do again. That's it.
Okay, well, my name is Nick Tober. I'm at the University of Exeter, and it falls to me to offer the vote of thanks. And I just wanted to say a few words about Pietro. I first got to know Pietro when he was a Royal Society University Research Fellow at Oxford, and we shared an interest in a class of proteins called hydrophobins, which are unusual proteins, natural surfactants. And we were both fascinated by those proteins, and that led to a collaboration, a very fruitful collaboration. And since then, we've collaborated on lots of different projects, including the barley powdery mildew project, and he's always been a great pleasure to work with. I know that his interests in biology go back right to his childhood, and he mentioned the fact that he was a budding zoologist until he realized what that might mean. And I understand from his family this actually meant a sort of passionate collection of animals of all different types. I understand he collected lots and lots of snails. He collected frogs. His father was a Baptist minister, and he actually managed to put frogs, I understand, in the font when he was a child. So this is sort of typical Pietro. Whatever he does, he goes at with great gusto. and this is his hobbies in sailing, I know his DIY work in the house, all of his hobbies, all of his interests, he goes out with great passion and great enthusiasm. And I think that you can tell that from his talk. You can tell that the same is true of his science. So what's Pietro like? What is he like as a colleague? Well, I just offer up 3 observations from the talk. The first is that Pietro took great efforts to explain everything to everybody who was here. Whatever their background, whatever their level, he explained everything that he did. And I think that says an awful lot about the man. That says a lot about his commitment to education, his commitment to being an effective teacher, and also to include everyone in this event. The second observation is this lecture theatre is full of his former students, his colleagues, and colleagues from lots of other different universities who've taken the time to come here. That also says a lot, I think, about Pietro as a collaborator, too. And I think the final thing is the fact that he thanked everyone throughout the talk, all the people that he worked with him, which also says a lot about him, too. He's a very gracious collaborator. So I really leave you with those 3 thoughts about Pietro. It's a great pleasure to be here. I'm very, very pleased that I could be here. He's got a very well-deserved personal chair. I'm very pleased for you, Pietro. Congratulations on that. It is well-deserved. And I'm fascinated by these Ralph effectors. I think they're phenomenal. So I actually think that all of your best work, actually, your very best work is ahead of you, because I think this is an incredible line of work to go down, because we really don't know what they do. We have no idea how they are subverting plant immunity, how they are affecting information flow within plant cells. So there's a whole wealth of things to discover. So I think this really is a rich vein for the future, which bodes very well indeed. The final thing I'll leave you with is the one thing I learned, which has sort of confirmed something I've long suspected, is that Imperial College professors dine every evening on grapes, which are fed to them on big white platters, and that's washed down with lots of red wine. I always suspected that was true, and now I know that it is. So you've joined a very good collection. So thanks very much. Congratulations.